That's Eliza. You're going to get started. I think there's a few more people coming along later, and I'd just like to welcome Seth. He's going to talk with us today, and on a subject that's really close to his heart. Mouse models of autoinflammatory disease. Thank you.
Yeah, so this is indeed something that's very close to my heart. We do learn a lot from these models, and I think what I'm going to show you is a whole bunch of human diseases and basically their mouse equivalents. And so a lot of it, there'll be a few. I know you guys are pretty cool with the mouse histology and tearing them open and blood and guts everywhere. But there's also going to be some photos of, sick kids, and, they're not great, some nasty sort of skin lesions and these sorts of things. So I'll sort of give you a warning when those sort of coming up. If you don't want to look at them, that's okay. But the good thing is that for all of those diseases I'm going to show you, we've got great treatments and great therapies. And a lot of those therapies are based, entirely from what we've learned in the mice. So I think, it shows you really the impact of the work we're doing together with you guys. Unfortunately, because of time constraints, Liana, we don't have time to talk about infections today. And we're doing some wonderful stuff with toxoplasma and some other things. Maybe you can get Chris Tonkin or someone to talk to you about that later on. We're going to just focus, though, on the autoinflammatory diseases. So these are different from your autoimmune diseases, and you're probably all really familiar with autoimmune disease, right? That's where your T cells and your B cells get autoreactive and they start attacking your body. These are different. This is the activation of the innate immune system. This is your neutrophils and your monocytes and your macrophages. They're primed and ready to go anytime they see infection. But in these kids that I'm going to show you, and in the mice where we've modelled these diseases, we're talking about mutations that have genetically activated these pathways. So there's no infection, but the body thinks there is. And so everything, all these innate immune cells, the pus you see when you cut yourself, those white blood cells, that's the sort of cells that are going nuts in these kids and causing inflammation. So I know it's a simplified science talk, but the reality is it's really complicated. But this is really the sorts of pathways we're interested in, and you don't need to know any of the details. But just some of the names that are going to come up throughout later on in the talk. One of the molecules, NLRP3, is one that we really work on a lot, and that's mutated in some of the families. Another one called pyrin is really important. It's also one that's mutated in a disease called familial Mediterranean fever. These all have syndromes in families. Some of them are like this one. It's a heterozygous disease, so two heterozygous carrier parents would have an affected child. In this case, it's dominant, so you can trace it through families through multiple lineages. The important thing is that there's lots of things that all seem to feed into one pathway, and this pathway is really important for the generation of a really potent inflammatory signalling molecule, something called interleukin-1. And this same protein complex also generates another cytokine, one of these inflammatory signalling molecules called interleukin-18. So they're the two sort of signalling molecules we're going to be interested in. And they're what we can block to try and treat these diseases. So, here's what some of the patients look like. So if you don't want to look at nasty skin lesions, you can look away now. But they're everything that ranges from sort of urticarial skin rashes, and this is a disease we discovered when I was working at the NIH. Some of the lesions are more sort of urticarial in nature, so that often their skin manifestations are very different. This is one of these diseases that sort of turns up on house sometimes because the skin rash only really appears when the patient gets cold. And so you can put them into the cold room, and then this develops really, really quickly. It's kind of a diagnostic test for something like that would happen. Would not happen for most people. But often these diseases are characterised by skin inflammation. And so this is one of the diseases we discovered. This shows you how effective the therapy is. After only 7 days of treatment, this child went from being terribly sick, had joint pain, the bones were really sore, and the skin obviously really, really unhealthy looking here. And all of that, starts to heal in just 7 days of treatment, blocking interleukin-1, blocking the inflammation. And that's based entirely from the genetics, what we know about the mutation, and our studies in mice, learning what actually fixes the mice. And so that's a really wonderful outcome, because without treatment, it's a fatal disease. These patients are all dying around 10 or 11 years of age. With the therapy, it's just a magic bullet, totally normal, totally healthy. It's like nothing's ever going wrong. In that particular case, the mutation was so directly in this pathway that it's just like they're missing something and we put it straight back in. It's like one for one. And that's why the therapy is so good and so effective. I'll show you a few more examples of that. That's really been our main focus, is these really clear cases of genes causing disease, great drugs and therapies that we're getting for these really rare cases. But we're looking for funding from granting agencies who say, well, okay, you're able to help, a handful of patients. We're talking about, 100 patients in Australia, a couple of thousand patients in the world, not having a huge impact on that sort of scale. But then it turns out that these same inflammatory molecules like interleukin-1 and these inflammasomes, these NLRP3 molecules that are mutated, they're implicated in lots of other diseases, things like atherosclerosis, Alzheimer's disease, you name it, they're really important. Inflammation is a basis for these really, really common diseases. And so what we've done is we've tried to make inhibitors, small molecules, and so this is one that actually Pfizer made to start with about 20 years ago. But they didn't know what it targeted, and so they knew, it seems to work on interleukin-1, but at that time, in 2001, they were like, well, we don't really know how it works, got no target. And so they stopped working on it. They didn't generate a lot of IP, or they did patent it, but it's now off patent because it was too long ago. And so we started working on some of these molecules. And what we did is we've got the mouse model of the same diseases that I was showing you in the kids before. So this is taking the exact mutation from the patients and putting it in the mice. It's actually much more severe in the mice. And so both of these are affected mice. But you can see that they're really runted. This is at around about day 9 or 10, and Rhiannon would be familiar with some of these mice because she was doing some of this work together with Kat. They're quite sick and quite unhealthy. What that means is that, they usually die around 10 or 11, 12 days of age, but actually their mums don't like them much as well because they're runted. And so we try and foster them out to 129s or valve Cs, which is it? It's the valve Cs because they're much better mums. And so when they die, they're dying because they're sick and unhealthy and not just because the mum's ignoring them. So we think this is really the underlying disease. And that small molecule, that drug I showed you on the page just before, that's the guy that's been injected with that drug. And really, for as long as we were injecting the drug, these mice were able to keep them alive, which was a really great result. This is just a small molecule. It's not like a biological therapy or anything like that. It's really something that's going in and targeting that. I mean, we're not totally curing the mice. This is their weights there. And so, Rhiannon or someone else, usually Rhiannon was weighing the mice out through these stages here. And you can see that their weights are really increasing as well, which is great. Not totally back to normal, but their weights are definitely increasing. So that's really, really good. This is actually cells from the human patients. And you don't need to worry about what the bands are. But again, we're seeing this drug really works in the patients as well as in the mice. So that's really good. One of the other things we're doing is we're taking blood from these mice, which isn't a lot of fun, and so we have to go in there quite early, during these early stages where we still have a comparison to make. So around days 6 to 7, we can take a small amount of blood from these mice, and then we can look at the levels of the cytokines, these inflammatory molecules going around. And the small molecule inhibitor we've got works really well here. I mean, you can see a huge decrease in these inflammatory proteins. And we've also got another mouse mutant where the activating mutation is in another protein, and it has no effect on that. And that shows you specificity. So this drug looks really, really specific as well. So actually now what we're talking about doing, and we're talking with the large clinics where they have the patients with the same mutations, is we're going to take this small molecule drug and put it into patients. And so that's a real, started in the mice, and the first experiments in living organisms were done by Rhiannon here. And now that's going to hopefully go into patients in the next year or two, which would be a really fantastic outcome. Because currently the drugs they're on, they're good, but they're not like they're injections that they have to have every day. This is a tablet. I mean, we were injecting the mice, but we could have fed it to them as well because it's orally available, so patients could potentially just take a tablet. And when we're talking about giving people a tablet, then we can start thinking about, well, maybe we can just give it to people who have other inflammatory diseases, your cardiovascular disease, your Alzheimer's disease, your diabetes. It could work in a lot of these things. And in mouse models, it's giving us indications that it does. So it could have some really broad use outside of these really rare diseases. So I'm going to tell you 3 stories. That's the first one. It's sort of small molecule inhibitors. I think it's got a really broad scope and it looks really interesting, and it's a sort of classic model of autoinflammatory disease. So some of the other work we've been doing, and it doesn't sound like an inflammatory disease, but one thing that's clearly been shown is that underlying obesity is inflammation. And so we've actually learned a lot by looking at different mice that are genetically deficient for different things. And this is one that lacks interleukin-18, and we didn't publish this story, but I think you can clearly see that this mouse is this is the knockout mouse, and it's much, much fatter than the wild-type mouse. And that also goes along with differences in when you inject it with glucose or you inject it with insulin. They don't really metabolize the glucose or insulin the way you'd expect, showing that they have these metabolic diseases which are the mouse equivalent of diabetes to a certain extent. But there's obviously a lot of differences between mice and humans, but as close as we can measure it, these look like a pretty good model for that. And so we were wondering, what generates this cytokine, and we think we had part of the answer because we made mice that lack this NLRP1 gene, and we think this is what controls that interleukin-18 level. And you can see here, you can almost genotype the mice, and Rhiannon's seen these guys, they're really, really fat. Huge amounts of this adipose tissue here, the visceral adipose tissue specifically. And you can see that just measured here, just taking it out and weighing it, that sort of thing. The subcutaneous adipose tissue also is increasing in size, and the liver weight's increasing, and I'll show you a little histology from that in a second. So there's lots of lipid getting accumulated into the liver. And what you see in histology, and I don't know how well it projects here, but the actual adipocytes themselves in obesity, they swell up. So they go from being small little adipocytes in the fat tissue here, and then each one just gets larger. So you don't increase the number of cells that you've got in that tissue. It's just that each cell actually increases in size, and that's what leads to this increase in the total size of that tissue. And so, we take blood from these mice, and we look at things like leptin, because that's produced when you eat a meal, and it's supposed to tell the body to stop eating and to metabolize all these lipids. And in these mice, it's really, really high, but it's not doing anything. So it's like they're resistant to that leptin signal in the same way that they're insulin resistant, like they've got more insulin there, but it's just not doing anything. And together with help from Danielle and Kat and Rhiannon, we've been doing things like challenging them with glucose. And again, you can see they really don't metabolize the glucose properly, so they have resistance to the glucose insensitivity. So they really do have some sort of metabolic disturbance. That would be a mouse model of diabetes. And if you look at this as the liver, you can see all these little circles here. These are filled with the lipids. They're filled with all of those things that should be fat and should be in the adipose tissue, and instead they're getting into the liver. And fatty liver disease is one of the big problems for people with sort of chronic obesity, and definitely we see that in these mice as well. And so those are mice on a high-fat diet. And so what we've done is actually we've tried a few different sort of feeds, and you might have seen these in the freezers downstairs. We made a diet that's really, really, we call it high-FRED or high-fat reduced energy diet. Super high in fat, but actually hardly has any energy. So they're eating all of this food and it's super high in fat, but actually doesn't have many calories because we've taken out all the protein, taken out all the carbohydrate, and actually we get rid of the phenotype. So even though the mice are eating heaps of lipids and they're ingesting heaps of fat, they don't put on any fat and they don't display the phenotype. Really, the phenotype comes because they're of the amount of energy, the number of calories they input. So in this particular diet, it's a high protein diet, and that diet, it's super high in energy and in calories, but it's got absolutely no fat whatsoever. And there, the mice get fatter, and you can see that they have increased adipose tissue and subcutaneous adipose tissue fat. When you inject them with the glucose, it doesn't get rid of the glucose over time, it maintains in their blood, so they sort of have these metabolic diseases that you'd associate with insulin resistance and diabetes. So there's a couple of diets that we've been playing around with. They're not the best diets in the world. It's really hard to specify exactly what constituents go into it because the guys who are making these are like stock feed sort of people. So they don't really have the exact, biochemical breakdown of exactly what's gone into these diets. And I think it'd be good to get some more of that sort of information. But it's really hard to get a breakdown from what they're putting in being really raw ingredients, not defined chemicals. And we could move to diets that are defined chemicals as opposed to raw ingredients, which might be something we try to do in the future. But I think that we've seen some pretty interesting results, and it's been quite interesting. And actually, you can see a lot of differences in the mice when they eat these different diets. And so I think that'd be interesting work to do on these things in general. So we've also got mice where we activate this pathway. So those mice are mice where we knock out the pathway and then they get fat and obese. So now we're activating this pathway. We're making increased levels of interleukin-18, and we were hoping from skinny little mice that were protected from obesity and insulin resistance. And that was exactly what we saw. So these mice are really, in terms of obesity, they're thin, they're healthy, they're happy. But then when we had them on a high-fat diet, all of a sudden they started to die. And I think you can see what's happened here. We've been too good. And there's absolutely no adipose tissue on that mouse at all. And so we've activated the pathway too much, and it's just burned off all its fat, and now it's starting to die. And that's really quite a remarkable finding. I don't really know of too many other mice where you feed them a high-fat diet and they drop dead. So that's really quite important to know if you're thinking about developing therapies to increase this pathway and fight obesity and insulin resistance and diabetes. You definitely don't want to push it too far. because that could be really, really bad outcome. And you can see some of the worst histology. We don't know exactly why the mice die. They just sort of see, I think Rhiannon's sort of seen them, they just sort of get listless and they just sort of sit in the cage. It's like they've just got no available energy, because they would normally get some of their energy from this fat and from this lipids, and they've just got no energy anymore, and they sort of sit in the bottom of the cage and they just lie there and waste away. But some of the worst histology we see is in the liver. So normally you get these little bits of lipid here, these little circles, and you can hardly see any of them here anymore. It's all necrotic, and the mice are really unhappy and they're dying there. So that's quite an interesting finding. All right, so there was the second story for you there. Have you got any questions? I do talk rather quickly. It's hard to get in, I know. You all following it okay?
Yeah.
All right. One more story to go. This one's really interesting and it's brand new, so it hasn't been published yet. So not many people know about it worldwide. But so the original observation was already published. And so this is going back to more that sort of classical autoinflammatory disease phenotype. And Ben Kyle was the person who originally found this mouse, not here in Australia, but actually in Baylor during his postdoc. Really classic inflammation just in the ears and in the tail. And you guys might have seen these mice around. They've been around Weehai for ages now, so many of you may have seen these mice. So that they can scab over and get quite severe, especially after clipping the mouse ears. And we've tried some experiments actually measuring the size of the clips and the little holes in the ear, and there seems to be some evidence that they don't heal normally, so that would be in agreement with these sort of scarring around the ears and things like that. So we think maybe there's a wound repair deficit, and that might be important, as I'll show you later on. So it was called red ears mice for an obvious reason, and that's an interesting sort of mouse model, but during the initial publications, we didn't really know the molecular basis for what underlies that. Although we knew it had something to do with actin polymerization. Actin is like one of the building blocks of the cell, and we know that it's really important for the shape of the cell and moving the cell around, these sorts of things. And these mice have way too much of it. The cells are full of actin because they can't get rid of it. And so the question is, what's all of this actin doing? And we've been attacking that at a molecular level. I'm not going to go into too many of those details with you. I'm going to try and stick with the biology of the disease. These are the neutrophils from the mice, and these are neutrophils from normal mice, and these are from those red ears mutant mice. And this is the nuclei, where the DNA is usually kept within the cell. And you can see, instead of being kept within the cell, this is really bursting out and exploding. And that's a really weird-looking neutrophil. So that's quite something that we noticed, but we didn't really mean anything to us, but it is going to be really important in a couple of slides' time. What we've tried to do is identify the cytokines and the different parameters that underlie this inflammation, so we measure all these different cytokines. But then we cross the mice to things that are deficient for all of these different things, all the same proteins I've just been telling you about, interleukin-1, interleukin-18, and we can see finally something that protects the mice. And it's the interleukin-18 molecule that protects the mice. So we're starting to get some sort of understanding of things that protect the mice in terms of the molecular basis. And we've gone on to try and get rid of the cell types that make the inflammation. Actually, the neutrophils don't make the inflammation in these mice. We've got rid of the neutrophils using depleting antibodies, and genetically we've also depleted them, and the mice are still pretty sick and have the inflammation in the ears and the tail. But what we did get success for is depleting this population of cells here. In fact, you can see that when we put in these things called clodronate liposomes, and Man Liang was doing a lot of these experiments when he was in my lab last year. And when we put in these clodronate liposomes, what happens is the monocytes and the macrophages, they eat the clodronate liposomes, and when they eat them, they can't get rid of them, they can't digest them, and then the cell just explodes. And so that's how we get rid of those cell types. And then, again, we really are able to keep the mice happy and healthy for as long as we inject them. Previously with the other molecule I showed you, and with this one as well, we do get some sort of, if we inject them too often, we do get an injection site reaction. These are IP injections mostly. And so that's just something we watch out for. I think some of these mice sort of got a staph infection after a while as well. We didn't know whether that was associated with the injection or just, because of the drug we were using or something like that. At any rate, we were able to protect them for while we were injecting them, but after we stopped injecting them, then they sort of got a bit sicker and we had to get rid of them. And you can see that here. These are the ones that are injected. They don't have the ear inflammation in them. Ones that have the ear inflammation just got the PBS control. So now these photos are, we're treating mice, and now I'm going to show you the humans. So these haven't been published yet, but there's a couple of families, and they have a really, really severe disease. It's exactly the same mutation as the mice have. And so based on the phenotype of the mice, we've got a therapy that works for the humans, but you'll see if you want to look, it's not a very nice disease. They have really nasty scarring around their mouths, and I'll show that to you now. It's, and it's a terrible disease and really severe scarring, and some of these patients need to be intubated because they just can't eat, and it's really a horrible disease. But you can actually see, like, the reason why we should have known about that disease was because this was the year I was born, 1978, 36-something years ago. A patient was described with something called lazy leukocyte syndrome, and that's the exact disease the mice have. Look at this neutrophil. It's exactly the same as what I showed you before. You can see the DNA bursting out of the nucleus, and it's exactly the same disease. So if we noticed this a few years ago, we would have put the puzzle together a bit earlier, but we got there in the end. And the way we got there was because our friends at the NIH were able to sequence the gene in those patients, and they found it was exactly the same mutation as the mice. So the great part about the story is that the mice, we can cure them now by transplanting their bone marrow. It's harder to do the therapy I was talking about with the collagenate liposomes, destroying the macrophages, which might work, but that's really not approved for human use yet. But bone marrow transplant works in the mice, and they've done bone marrow transplant, and all the kids have done really well after bone marrow transplant, so that's a really good outcome. And because of their neutrophils not working properly, they get horrible infections, and the mouth, the stomatitis that I show you, really terrible. But after the bone marrow transplant, all starting to fix up, all getting a lot better. So a really good outcome for those patients. To be honest, we're learning so much about this actin protein and the things that recognise and all the stuff that's going wrong there from this mutation. It's scientifically astounding, but to be able to be making some actual impact on patients' life as well is a great outcome for those patients, so that's really good as well. So, look, there's a summary there, but the important thing is to thank everyone that's been involved. Huge amounts of work, and I hope that the gold here shows you the importance of the mouse room staff, and my apologies if I've left anyone off, but we've had so much help from you guys, and it's been really fantastic working with so many of you. I really think that we're really lucky because a lot of these studies we do are the direct correlates of diseases in humans, and the therapies that are potentially out there when we work out what's going wrong are really, really great. So we're really, really thankful for all of your help, and we really enjoy working with you. So thanks very much.
When you're using these therapies to suppress a certain part of the immune system or the inflammatory response, what happens when you're targeting one sort of disease? What happens if, say, the person gets a bad cut and gets infected? How does that affect?
So it depends on the drug. Actually, these drugs have fantastic safety profiles. So it's very rare to see infection. But some of the longer lasting drugs, the biological therapies that target a particular cytokine, you can stop taking them, and it'll depend on the half-life of that drug. One of the drugs has a half-life of 24 hours, so that's great. If you do get an infection, then you go off the drug and you can get rid of the infection very easily after you get off the drug. Some of them have a half-life more like half a month, and that can take longer. But as I said, the safety profile is generally pretty good, and that's because you're targeting one thing specifically. And our human response to infection is with a broad array of things. And if you just target one of those, usually the infection's covered by another part of that. And so we just have to be, we're now thinking about targeting multiple things to fight inflammation. That's more dangerous in terms of infection. So it's a double-edged sword. We have to be careful how we play it, but most of the time it seems to be okay.
Yep. And are these treatments would be ongoing through the patient's life?
Yeah, so there's an amazing talk going on today at lunchtime about CRISPR technology and genetic editing. I mean, that's the next frontier. It's a bit of an ethical minefield, but you can certainly imagine that for these sort of mutations, if you just went in and corrected that genetic change, not only would that person would never need another drug or another therapy their entire lives, but their children will not have the diseases either. But it's a real minefield. Do we go and do we mess with people's genetics forever? It's one of those really, really difficult questions. The awesome thing in this particular case is we've got drugs that work, and so we don't have to deal with that issue. But there are certainly some of these diseases where there are no known drugs, but we do know the mutations and could potentially fix them. So it's a really interesting question.
it's really interesting talk, but I'm interested in your high-fat, low-calorie diet.
Oh, yeah. I can't tell you too much about it, except that that diet is probably quite high in fibre as well, actually. So you've probably actually got a couple of things. It's a high-fat, high-fibre diet. And if you look at these mouse cages, the bottom of it is just full of poo. So I think they're eating a lot of fat, but it's just going straight through them. So if you're thinking about taking on that sort of diet, I think you'll need your own personal bathroom. But you know, those mice weren't getting that fat, so maybe there's something to be said for a high-fibre diet, you know. So. All good? Thanks, guys.